The MUHC Human Islet Transplant Lab is a state-of-the-art facility that provides highly purified preparations of insulin-producing islets. We receive roughly 30 pancreases a year from multi-organ donors. A matched donor pancreas to a recipient is used for islet transplantation. Pancreases that have only received research consent are isolated and cells distributed to diabetes researchers around the world. Pancreas biopsy samples are obtained from pancreases that do not meet our strict islet isolation selection criteria. In each one of these scenarios, we obtain many tissue and cell samples that need to be properly stored and organized for future research purposes. We currently have about 3,400 samples from 110 donors. Prior to receiving our biorepository in a box, as you can see here, we used Excel spreadsheets to keep track of inventory, sample location, and distribution. This primitive method of organization actually made it difficult to streamline cell and tissue processing for biobanking. Aliquots weren't getting entered quickly enough, and data wasn't being updated with the number of samples remaining after distribution. An alternative method needed to be devised. That's where the Canadian Biorepository software comes into play. It's a great tool that allows us to keep track of patient information, sample inventory, location, as well as distribution. So one of the first steps after processing our cells and tissue is to create labels. Each one of these labels is generated using a unique identifier code. We can then assign other identifications such as study sample ID, as well as the collection date and the processing date, and enter other data fields. Once we've entered all of that information, we can then simply print off the label. Once printed, the label is simply affixed to each sample tube. So barcodes are on the label itself as well as on the top of the cap. So after putting the labels on our tubes, we then use the Canadian Biosample Repository software, which will enable us to enter information about our patient, or our donor in our case, as well as information about the exact samples that we are collecting. So currently one of our studies is obtaining pancreas biopsies. So we've created a study in our software. All the information that's entered in the software is stored on the servers, so nothing is stored locally. We also assign different types of aliquots to our study that needs to be processed or that ends up being collected. We also generate different container types which end up showing the location of our samples in our -80 freezer. So once we created a study, we have to generate a patient, or we enter patient information. We then generate a collection event as well as then a processing event. After creating here are given an example of a patient that we entered. We assign our own codes to all of our donors. So for example, in this case, HP 2015-02. So create a collection event here, which is the time of cross clamp. we then ended up creating a processing event, which is the time that we received the pancreas and processed it, and then obtained the various samples, which are stored in our -80. So after creating the collection event, we create the processing event. So like I mentioned earlier, the processing event is the event at which we received our donor pancreas and obtained our various biopsy samples. Once we obtain all of our biopsies, we then need to link those samples to the labels or the tubes that we put them in. So this is called the specimen link. So here we're just simply linking that sample tube to a specific to the specific sample. For example, formalin. A formalin sample, for example, that we have obtained. One of the ways of doing this is by simply scanning the barcode and then entering that information in that specimen link, or if we have a large a completed box with all the sample codes, you can simply just take a picture of the barcodes and then let the software identify what the barcodes are, and then we can link those barcodes to the actual specimen. So this is called decoding the palette. So this is where we took a picture of that box and then letting the computer software decode those sample barcodes. So using the software, we can simply then link all those barcodes to the sample that we got. After we linked all these sample specimens, we need to assign a location in our -80 freezer, and this is simply done by linking or assigning each of those tubes to a specific position in a 81 well box, for example. Another thing that we can also do, which the software allows us to, is then export some of our samples to other labs, or distribute our samples to other labs. It's pretty much like a shipment, pretty much trying to ship and receive samples, and in doing so it helps us keep our inventory updated. So once all our samples are linked and assigned, we simply just store them in our -80 freezer.